So my name is Tereza Manousaki, and I'm a researcher on the Hellenic Center for Marine Research in Greece. My research focuses on evolutionary genomics and on how genome evolution is linked to species phenotypic evolution and diversification. I have followed the genome papers from the era of the Nature paper until the era of the genome node, and I have been really amazed by the progress of the field. On working with genomes, I have been involved on different aspects of downstream analysis, such as population genomics, aquaculture genomics, comparative genomics, and pipeline development. So it's really an amazing time to be working with genomics, with genomes. So ERGA has been really fundamental in spreading the use of genomics to a really wider community compared to before. It had shared knowledge and resources with all countries of Europe, regardless of the existing capacity and infrastructure. And it has been an example of collaboration at the scientific and at the European level. Now, based on my background, Data Analysis Committee is one of the most important committees because genome reference provides the basis for a number of downstream applications, which lead to a deeper understanding of the species, biology, and evolution. What ERGA Data Analysis Committee has done since the beginning was to launch questionnaires trying to map the community's interests. So also since middle 2023, we launched a very interesting seminar series called Biogenome Analysis and Application Seminar Series, jointly with Biodiversity Genomics Europe. This seminar series is focused on the use of genomes in the areas of population genomics, phylogenomics, comparative genomics, and functional genomics, which are the four sub-areas that DAC is focused on. And what is interesting about our seminar is that it has the main presentation of the scientific question and how it was answered. And then it has a second session which dives deeply into the problems and difficulties that the scientists faced to answer the question. So it has a much more focus on the actual analysis that took place. We have also started community meetings, which have gathered a very diverse group of scientists participating. And we aim, on one hand, to build a guide with solutions to the most important downstream application of genome references. And on the other hand, what we expect from our community meetings is to identify interesting topics that the community would like to work on and build something on them. So the most interesting is at the same time the most challenging part of ERGA Data Analysis Committee, and that is the heterogeneity of the members that are part of the community. It's an amazing privilege to have in the same room or working group people that are working on conservation, on comparative genomics, on phylogenomics, and on functional genomics. And this is the great potential that we need to take advantage of. Okay, what I'm really excited about is the number of genome references that are being publicly and openly available by large consortia. And there is an amazing feeling with genome references of the tree of life, bringing biology to a new era and making us reconsider the term non-model species. What is also really interesting is that now species that are really rarely studied or strange organisms are being sequenced. First, to offer best practices for genomic downstream analysis. And the second would be to engage the community, identify gaps in the four particular areas we're working on, and then work all together to fill them. So thank you very much. I hope that we'll see you in one of the next DAC meetings.